Hey, this is Mr. Mitchell with another video. You know the drill about the questions. Go ahead and write them down. Let's talk a little bit about a couple of different things. We're going to talk about meiosis, and then we're going to talk about DNA. We talked about how Mendel in our last video proved that there are genes and chromosomes. In 1903, this guy right here named Walter Sutton, nice hairdo there, Walter, took it farther. He was studying, of all things, grasshopper reproduction. Now, what Sutton discovered was something strange about the sperm and the egg cells, the sex cells. The sperm and the egg cells have half the number of chromosomes as the regular cells. All the other cells in our body have those 23 chromosomes which replicate to form 46. But that's not the true for sperm and egg cells, and that's not true. That's also the case for other organisms too. The sex cells have half the number of chromosomes. When the two cells merge, the chromosomes combine to form the regular number. And I've got a couple of diagrams here which will show you a little bit about it. But you know how we have these, we studied last time, we had these little cross-like things. Those were the chromosomes there. And in this process, it actually breaks down to an individual strand, not just a strand that is an X. So that means half of the chromosomes you would normally have. And that process is called meiosis. We called something else mitosis. Mitosis is that process of dividing the cells, that middle stage. Meiosis is the division of these sex cells. And what they do is something different. They end up dividing not just into the X little chromosomes, but to just a single strand, and those single strands will end up combining to make the new, in the new organism, you might say, or to assist in the new organism. So to reiterate, the meiosis is the process by which the number of chromosomes is reduced to half from the sperm and the eggs. Meiosis is similar to mitosis, except the chromosomes split twice. Instead of becoming two daughter cells, like you would see here, they become four. They become four daughter cells. Now, chromosomes are made up of many genes joined together, kind of like beads on a string. And if you were to take one of these over here, I'm going to try to make it a little bit bigger. If you were to take one of these right here and have the little centromere in the middle like you did before, there are thousands of genes on this chromosome, and just kind of all in order down a line, and there's no possible way I can get thousands on here, but there are thousands of them on this little chromosome right here. Now, for each one of those, they all have genotypes. So I'm just going to use some examples. Let's say this one right here is eye color. It has the genotype of, well, we'll say this is the male chromosome. Let's say it has a eye type of blue, which would be dominant. And you may have over here the female chromosome, which would have the one for eye color. And let's say it would be something like hazel. Either way, it would not be dominant. So all down the line of these thousands of genetic genes, these thousands of genes on this chromosome, you're going to have the genotypes. You're going to have like big B, little b. You're going to have like a capital R, capital R. You'll have all those different genotypes that we studied about with Mendel. And it's just amazing how many of those our body processes through. And that's exactly what happens whenever we do have a new organism. You have all of these genotypes, which end up, you could calculate in a Punnett square what they would end up being. It's just more than the mind can even imagine sometimes. So now we're going to talk a little bit about DNA itself. DNA and chromosomes are mostly composed of DNA. These right here and just the regular DNA where we're talking about. We're going to kind of skip past meiosis and how we get the DNA to making proteins. How we get the DNA to making proteins. DNA is made up of 4 different bases. You may remember it is a double helix. It is located inside the nucleus of all our cells. And these bases here are made of nitrogen bases, and they're adenine, thymine, guanine, and cytosine. You can see they have to be partnered up with the right ones or you have some kind of mutation. Now, this DNA carries all the chromosomes, all the genes that is in all of our cells, not just those sex cells, but in all of our cells. And the order of these bases makes up what we call the genetic code. The genetic code is the order of all the adenines, thymines, guanines, cytosines, and so on. You could literally list them back to back to back to back to back, and that would be our individual specific genetic code. The genetic code that we have and that all organisms have determines what proteins are produced. And the proteins, now keep in mind, all of this is inside the nucleus. We're just still in the nucleus here. The orders, I guess you might say, come from the DNA, from the genetic code as to what proteins are produced that produce the enzymes, that produce the actual phenotypes that we have. So the chromosomes are inside the nucleus. The question is, how does the DNA protein get outside the nucleus? Well, the answer is going to be here. It's going to be RNA. We have 2 different kinds of RNA. RNA takes it from the nucleus to outside the nucleus. Now, I've got kind of a small picture here, but you see we have DNA here that is inside the nucleus. And they have RNA, which is going to be 1 strand. It looks like half a DNA, you might say. In fact, you can see it right here. RNA is just the 1 strand of the double helix, whereas the DNA is 2. So that's going to play a big part in how it gets out. Now here's what happens. You have the DNA which comes unzipped, purposely comes unzipped. And what happens is the RNA comes in and replicates, gets the messages from what the DNA is saying. So the messenger is the messenger RNA. This is a messenger RNA right here that actually goes into the nucleus to get the information from the DNA. So let me say it again. You have the DNA that unzips. You have the messenger RNA that I guess you might say plugs in to the individual strands, to the adenine, the thymine, and the guanine, and so on. After it plugs in, it will then leave the nucleus. It will plug in and get the information, and it will leave the nucleus. Notice here it is leaving the nucleus. And in the nucleus, it's going to attach to a ribosome. You may remember the ribosomes. We said that the ribosomes have to do with the protein. And I'm going to spend so much time if I write out the ribosome, but it's going to be ribosomes that I would end up writing out right here. So that goes to the ribosomes. Now, at the ribosomes is where you're going to get the transfer RNA. So the transfer RNA will attach to the messenger RNA. The transfer RNA looks like this. Now, it's kind of a long story as to why it looks like this, but you can see that it attaches to these plug-ins, you might say, from the messenger RNA. So once it attaches, information is transferred, and the amino acids, now remember, amino acids form proteins. The amino acids form a chain, and you might can see a little bit of this chain right here. You can see it bigger right here. We have the chain, which is a protein chain, coming from the ribosomes. You have the messenger RNA, which is there. The transfer RNA connects to the messenger RNA, and it brings the proteins, it brings its proteins and makes a chain with it. The chain grows longer with each RNA and transfer RNA, and then the chain is released into the cell, into the cytoplasm, and it will get more amino acid. So that is how we get the genes, the premeditated phenotypes, or I guess it would be the genotypes that produce the phenotypes, which actually comes out into our cell. And from there we end up being who we are with all our individual intricacies. If a mistake occurs, you have mutation. Mutations cause a cell to produce incorrect proteins, and the phenotype will be different as a result. Now, the mutation of a body cell is not passed on to the children, but if you have a mutation of a sex cell, then it is passed on to the children. Mutations could be several things. It could be the wrong nitrogen nitrogen base paired up. It may happen as a faulty replication. Let's say this doesn't replicate right. You could even have incorrect mitosis, and that might cause too many or too few chromosomes. Mentioned earlier that Down syndrome is an instance like that, where you have multiple chromosomes of, I think it was the 21st chromosome. But anyway, not all mutations are harmful. I mean, there may be mutations which really don't have much of an effect, and there may even be mutations which help an animal. But here are some internet pictures I found of mutations. You can see this frog with multiple legs. You can see this turtle with 2 heads. And you can see this hat, which has this, not hat, but cat, that has all kinds of toes right here. Well, it's a lot to swallow here, but we have meiosis and we have DNA that I hope you're able to get a lot out of this video. Don't forget to go back and do the questions.